So we're going to go ahead and start. I just wanted to let everyone know we're recording. We're going to ask everyone to please stay on mute. We'll have a question and answer period at the end. So I've asked Drew to go ahead and just move through his complete presentation first so that we can hear all of the rich information that he has for us. So yeah, I'm Donna Ball. I'm the lead scientist for the South Bay Salt Pond Restoration Project. And so thanks today for all of you for joining us. I'm super excited to welcome Drew to this monthly speaker series. He's the treatment manager for this State Coastal Conservancy's Invasive Spartina Project. And I'm not going to go into his deep experience in wetlands and invasive species management. You can see that in his bio online. But I just want to share that I've been really fortunate to know Drew for almost 20 years as he joined the Invasive Spartina Project about the time that I started to work on marshes in San Francisco Bay. So it's been fun to be a colleague all these years, Drew. I
Yes.
just want to say that the rest of he and the rest of the staff at the Invasive Spartina Project, I have to say they're some of the hardest working marsh ecologists in the estuary. They're out annually surveying and treating hundreds of miles of marsh in search of invasive species and installing plants in areas where the invasive species have been controlled and in monitoring species that use those marshes. So super excited to have you here today and to hear about all of that work. So welcome, Drew, and I'll turn it over to you to tell about these three species in particular that the Salt Pond Restoration Project is tracking as part of our adaptive management plan.
All right, great. Thanks, Donna. Yes, it's a pleasure to be here with you all today. And I gave a presentation at the 2022 Science Symposium for South Bay Salt Ponds and had the same start of my title, Breach It and They Will Come. And I just couldn't pass up using it again because I thought it was so witty, I must admit, lacking humility. But it is true that that we have a lot of joy in seeing these marshes develop, but we also have a lot of work. It brings a lot of work to steward them and to it can't happen in isolation. It can't happen in a vacuum. So here's a short presentation map. I'm going to cover 3 non-native invasive plants that threaten various zones of our tidal marsh ecosystems in San Francisco estuary and within the South Bay Salt Pond Restoration Project. I want to note that these management efforts for these 3 noxious weeds are very separate from one another, although they often use same team members, and we're working in the same general footprint. They're funded differently, permitted differently, and such. So we're going to talk about the hybrid Spartina, the hybrid between Spartina alterniflora from the East Coast and our native Spartina foliosa cordgrass, and the Coastal Conservancy's Invasive Spartina Project, as Donna mentioned. We'll talk about 2 newer invaders, seashore Paspalum, Paspalum vaginatum, and invasive sea lavender that we've been working on, and many volunteers have been working on for quite a few years in San Francisco Bay. I was fortunate enough to be at the Mount Eden Creek Marsh Breach 2008. It was a day off from treatment at Spartina Project, and it's just very exciting to see these areas opened up. To think of the potential of new marsh acreage and all the habitat it's going to provide, and the wildlife it will support. But at the same time, as these breaches bring in the sediment needed to create these marshes and the native plant propagules to start to populate them and create a native plant assemblage. They also can bring in the non-native plants and the invasives. And these areas, when they're first opened, are very vulnerable to invasion. They don't have what we call biotic resistance. They don't have the dense native plant cover already established that can help them to resist invasion from some of these very aggressive plant noxious weeds. So that's when they're most vulnerable, and we need to protect them and allow them to develop on the trajectory that we hope that they will, to provide that habitat. And since 2006, thousands of marsh acres have been added to the Invasive Spartina Project's annual monitoring and treatment work area. And these areas take a disproportionate amount of work to monitor, especially as they develop and really get an extensive native plant community. And they take a lot of time away. So they really add a great burden, but they bring all the benefits with them as well. So we care a lot about them. We want to prioritize them, but they do add a lot to our plate. So The Spartina that we are going to be talking about today is the hybrid Spartina between the East Coast Spartina alterniflora that was introduced in the 1970s that hybridized with our native Pacific cordgrass Spartina foliosa. The UC Davis Strong Lab and others have done over 20 years of research into this hybridity and produced over 2 dozen published papers on it. You can see on the left the general range of our native Spartina foliosa kind of plays well with others, stays within its lane, goes down into the low marsh, forms the lowest vegetated emergent vegetation in our tidal marshes, just above the mudflat elevation, and then it grows up into the and shares space with the marsh plane and the pickleweed and other native marsh plants. Whereas the hybrid has a very wide range; it goes well down into the mudflats. Converting those mudflats, creating a type conversion from those mudflats to a vegetated state that basically removes some of the benefits for shorebirds, makes in some of the invertebrate communities more inaccessible and changes the entire invertebrate community within those mudflats. It also grows through the mid-marsh in a very dominant way and excludes other native plants and can grow even up into the high marsh. The hybrid's often characterized by greater size, but it's a hybrid swarm that back crosses and is constantly creating new forms, new morphologies, new phenologies. So it's quite a complex thing, but most of them are larger, and that also helps them to grow down into the mudflats and send oxygen down into the roots from their parenchyma. The hybrid can pollen swamp the native. And this is one of the biggest problems when it gets into these new restoration sites that are starting to have a wide cover of Spartina foliosa. A little bit of the hybrid can pollen swamp the native, forcing it to become a factory of hybrid seed production, which really facilitates and accelerates the spread and can fester in these areas undetected while it's still creating hybrid seed. So there's a great shot of the pond A20, one of the island ponds, in 2019. It was breached in 2006, and this is how widespread native Pacific cordgrass Spartina foliosa is in that area. Beautiful. Very, very, very little hybrid cordgrass in there. So why is hybrid Spartina a problem? It obviously displaces our native plants, as I mentioned, and that degrades our tidal marsh ecosystem, reduces biodiversity. It dominates the mudflats, and this is something I keep trying to drive home time and time again in my presentations because I think it's lost on the average person. You see a mudflat, you're not really seeing the thriving community that mudflat supports. The shorebirds, the millions of shorebirds on the Pacific Flyway, our resident shorebirds, they need those mudflats to be unvegetated in their natural state. And when the hybrid takes over those, it converts them, and that's a negative thing, alters that entire benthic invertebrate community. It also threatens native tidal marsh restoration by coming into the sites early, dominating them, and excluding native plants from developing and developing on their native trajectory, finding their place within those tidal marshes. Eventually, it can endanger native Pacific cordgrass. Native Pacific cordgrass still represents over 99% of all the cordgrass, all the Spartina within San Francisco Bay, thanks to the efforts of the Spartina Project and its partners over all these years. Were able to stop that from being a factor. It can reduce flood control capacity by growing deep down into channels. That can happen for both the man-made flood control channels, but can also happen within our marshes and change the hydrology of the marshes by clogging these channels and starting to accrete sediment and changing their hydrology completely. So that's a very important factor. And it can also create mosquito breeding areas. We have some mosquito abatement groups are part of our partnership. So the Invasive Spartina Project began in 2000 as a partnership between the U.S. Fish and Wildlife Service as the federal lead and the California State Coastal Conservancy as the state lead. Treatment began after all the extensive permitting, EIR, EIS, in about 2004 with some pilot projects, and then really ramped up in 2005. And that's also when I joined the Spartina Project from my work up in Washington State as an aquatic noxious weed specialist with King County Department of Natural Resources. Hybrid Spartina has been reduced by our amazing efforts and our good fortune with funding and the support of the Coastal Conservancy and U.S. Fish and Wildlife and California Department of Fish and Wildlife, by about 97% estuary wide. Still a great deal to do, but 97% is an amazing thing. We have of our 221 ISP sub areas, we call them our sites. We have about 158 that now contain less than 10 square meters net cover of invasive Spartina. It's a little higher if you consider the don't constrict it to the space, the net cover, but the treatment cover. But that's a combined total of about 207 square meters. 56 of those sites were zero detect last year, and another 55 of them have less than 1 square meter of hybrid Spartina remaining. This is sort of a progression of heat map over a 5-year periods, where you can see on the left in 2005 how much of the San Francisco estuaries marshes were in the red or orange, very hot, had very high infestations. At least an acre of net cover or more. Start to get a little cooler in 2010. By 2015, you see quite a bit of green throughout the estuary, and that continues in the last larger map on the right for 2020. And then we'll update that again coming up soon here. The progress of the project has been slowed to some degree by a very careful approach to protecting Ridgway's rail, the endangered bird from in San Francisco Bay. And in some areas, we've done a very phased treatment approach, phasing in treatment over time to try to reduce impacts to the endangered bird. And as predicted in our EIR, EIS, removing hybrid Spartina monocultures that were so widespread throughout the entire bay, especially in the central and south bay, in this urbanized system where we had pushed our marshes right up to the very edge of all this development, did reduce the artificially dense cover that was present for rails, with this hybrid Spartina growing across the entire marsh and producing very tall biomass. It was artificially raising the cover for the rails above what it was in a normal marsh. This is much more problematic in fragmented marshes and those with low biodiversity than, say, in the refuge, in the Don Edwards refuge, but it is certainly a true fact across the marshes. Treatment was restricted at a subset of sites in 2011, 2012, in our biological opinions, to proceed with caution and not risk any jeopardy for the rails. Unfortunately, in the absence of management, some of those infestations sprang back to pre-treatment levels or much worse. We in 2000 we had a number of years where there were a series of sites, about 12 remaining sites after 2012, 12 sites mostly relegated to the East Bay that were restricted from any treatment or basically restricted from treatment, from full treatment. In 2018 biological opinion, the dark green sites on the map became part of our phase 1, and we were able to resume treatment at those sites. And by 2022, 7 of those 10 sites had already had greater than 94% reductions. And then in our most recent biological opinion in 2023, last year, we got permission to start phasing in treatment at the treatment initiation at the final 6 sites that are the orange sites on the map. We resumed treatment at 3 of those marshes in 2023, and by this year we saw some pretty amazing reductions at those marshes. These are marshes that are all hybrid. The native cordgrass have been completely assimilated by the hybrid swarm, so we're treating all the cordgrass in those marshes, and you can see some of those reductions there at the bottom line. Our biologists survey up to 40,000 acres of the San Francisco Bay marsh and shoreline each year. The project area is about 70,000 acres, but a lot of that is in that extra acres is in the far North Bay, where there's very, very little hybrid Spartina, very, very little invasive Spartina. We do this monitoring from June to November each year, and that informs our Spartina treatment program that I manage. We also do extensive genetic testing each year, sometimes 200 to 300 samples. This year, actually, a few more than 300 samples, tried to get a little bit more of that sampling to try to detect any cryptic hybrids, we call them, hybrids that are very difficult through their morphological characteristics for us to discern in the field, even our expert biologists. And this helps to preserve native Spartina foliosa while rooting out all the hybrids. I say, can you spot the more robust, bright green hybrid growing amongst the native Spartina? She can. That's our restoration program manager, Jeannie Hammond, right there. So the the task of going around all those 40,000 acres is pretty vast and it's hard to capture in a PowerPoint slideshow. But you can see two tiny little biologists on Greco Island combing that entire island and all the other marshes of the bay over the course of that fairly compact season to try to root out the hybrid. So you have all the challenges of of finding and discerning the hybrid from the native when it's not an obvious form, and you have all the navigation challenges of going through the entire set of marshes each year. The native cordgrass is actually more diverse. It's kind of considered genetically not that diverse, but in reality, morphologically, it's quite diverse around the bay, mostly related to the tidal amplitude around the bay, differences from the far South Bay and North Bay to the Central Bay. We have significant regional differences in size and other morphological characteristics like the length of the inflorescence, the flower on top of the plant. It can make hybrid ID very challenging. It often requires the plants to be mature, either within the growing season, later in the growing season when they're already flowering, or even it may take several years for some of the hybrids to develop. They're a clonal plant that's spreading out from a central point, or kind of radiating out. And as they kind of reach a critical mass, they often start to show some of these morphological characteristics that help us discern them. We can take DNA samples on ones that we suspect, but we can't do that across all the 40,000 acres of the bay. So. And then, as I said, I manage the ISP's treatment program. We have very narrow treatment timing windows that for most of the marshes around the bay, that are especially the ones that are inhabited by ridgeways rail, that we try to towards the tail end of their breeding season, we're allowed to start coming in and doing treatment, but we need to fit that in between all that mapping and treatment before the Spartina starts to senesce or die back for the season. It's a perennial plant. It dies back at the end of the season to its perennial roots and then reemerges in the spring and grows throughout the summer and autumn. So we have a pretty tight timing window to fit balance all these things out. We do very targeted spot treatment in most of the marshes, minimizing the impacts to the habitat. This is one of the first days we ever used airboat for treatment in 2008, and that's all hybrid Spartina in the photo there. So it's we've gained the luxury of being able to do spot treatment in many of these marshes now because of the hard work we've done over the years. And airboats are really essential to our access. We had no idea early on in the project how essential they would be, but we really want to get out to these sites as the tide is leaving them, really when there's no water present to treat them. And those are very narrow windows that even further constrict when we can do this work each season. The majority of the sites are treated by backpack. Larger infestations are still treated by hauling hose out from the airboat or for truck. That airboat is not stuck. It is actually where it's supposed to be, but I show some of the photos of that because it's pretty amazing to folks that aren't out there seeing this, that the things that our airboat pilots can do. That's Edgar from Solitude Lake Management, who's the best airboat pilot in the bay. I just want to bring up one challenge, one specific challenge. There are many challenges to our success, including the timing windows and all that I mentioned. But we've really found that there's a major significant challenge with drought versus rainfall. I call it the Scarpus effect, and that's from the old name for bulrush, the old genus name of bulrush, which is now Bulbochanus. It's our native bulrush is a brackish plant that cycles with rainfall and freshwater flushing, and in areas of the far South Bay and North Bay where it's more brackish, you will see it be completely absent above ground for years during like drought conditions, and then when rainfall comes, scores of acres emerge, dense acres, 6, 7 feet tall sometimes, emerge from the perennial root structures. Well, Spartina is not a brackish species as much, although it can survive into more brackish conditions. It's a much higher salinity plant, but it also can remain alive below ground, producing no to little above ground biomass, sometimes for years, especially during multi-year drought cycles. And that can thwart our efforts and our control, our detection, as well as any treatment. So the higher rainfall also, though, stimulates the emergence from the rhizomes of the cryptic hybrids. They will show more in our terminology. They'll perk up and display the more recognizable morphological traits in rainy years. So we may think that a stand is native, like the one in the photo here, and then it's hard to tell, but it's much taller than the native cordgrass around it, and much more robust. And during a rainy year, all of a sudden, oh, whoops, that was one we should have maybe been managing earlier. So And with no above ground biomass, mapping or treatment is really impossible. So we need to recognize those balances. We have an extensive restoration program, as I mentioned, run by Jeannie Hammond for Olufsen Environmental. We've been doing it for 12 years. This will be the 13th year. We focus on components of ridgeways rail habitat and increasing that. In a lot of places where the Spartina infestation was really large, it extirpated not only the native cordgrass, but excluded the native cordgrass, but also excluded things like the gum plant that's in the picture on the right. That's incredibly good rail habitat for rails to use for that they can build their nests in and use as high tide refugia. So there weren't propagules in a lot of these areas, especially of the East Bay, for the natives to come back in and repopulate after the hybrid Spartina had been controlled. So reintroducing those, we've planted over 600,000 of those plants at more than 40 sites over the years, and also constructed high tide refuge islands, partnering with H.T. Harvey on that, 72 of those at 16 sites, and starting to reintroduce the native Spartina in areas like Eden Landing is one of the biggest ones. And this is a shot of Eden Landing at North Creek Marsh in 2011. We had a small infestations of hybrid crowgrass there, essentially got rid of it by this point. And then we went in and planted heavily the Spartina foliosa over a number of years, genetically testing the plants, growing them out in beds so that we wouldn't have to damage intact marshes from removing too much of it. So we would take a few propagules, 300 or so, then grow them out in these large beds at the watershed nursery, and then outplant them. And within a few years, this is the kind of marsh that developed once the propagules spread out and found their own place within the marsh. And the first Ridgway's rail was detected in 2018 at this site, which was probably much faster than would have happened from its breaching to rail habitat without the assistance of active revegetation planting. And by 2021, there were 13 Ridgway's rail detected in this site, and I believe that was even higher last year in 2024. Spring 2024. And one more slide about the Spartina project. This is one of our best examples from the old days. I wish we had drones in the old days. I did get to take a lot of helicopter shots in the old days, but in the beginning of the Spartina project. But if we had drones, we would have had a lot of fun before and after shots. But this shot shows kind of what it's easy for folks to forget, the scale of the monocultures that were created by the hybrid Spartina and the threats that they posed once we've eliminated them. And South Bay salt pond sites are open mud and vulnerable in their early stages, just like the Alameda Flood Control Channel was. This was the original introduction site for the Spartina alterniflora in the 1970s. You can see in 2003 on the left-hand side, these clones starting to move out across the mudflats. And by 2005, just 2 years later, you can see these dense meadows of very large proportions had established. Over many years, we got rid of the hybrid Spartina at that site and started to introduce the native cordgrass. You can see these plots of us planting. And by 2019, we have a beautiful band of the native cordgrass at its proper elevation. We have pickleweed and native cordgrass spread throughout the mid-marsh elevation as it goes up higher, and a much more thriving community. This is a flood control channel, so it's not the same quality habitat as a Don Edwards Refuge, more intact marsh. But you can see this is a great example of the full cycle from hybrid infestation to revegetation to habitat, to stable habitat. And most restoration in San Francisco Bay relies on passive native vegetation. This is Knaptrack down on the mouth of Aviso Slough when it's kind of finally reached the point of what we call it's a marsh. It has marsh vegetation widely spread throughout, and it really requires ongoing stewardship to monitor these sites. As I said earlier on, this is one of these sites are the most vulnerable to invasion at the early stages. And once they develop this native plant community, the time burden to effectively monitor them and potentially treat if necessary really increases exponentially, because you can't just drive an airboat out across a mud flat, a mud plain, and find everything that needs to be treated. You have to search all the little nooks and crannies over the course of 100 acres of marsh, so it becomes very time consuming. So I'm going to switch gears to Paspalum vaginatum, one of the other plants I'm going to discuss today. It is called seashore paspalum or Paspalum vaginatum, and this is a chronology of kind of an relatively early detection and rapid response for it. I happened to see it called out in the annual weed alerts at the 2020 Calypsy Symposium, California Invasive Plant Council. We subsequently field identified it and confirmed it. Simon Gunner at OEI, our best botanist for the ISP. And this started to generate conversations I had with relevant landowners and managers. Luckily, the South Bay Salt Pond Project stepped up with some interim funding in 2021 and 22 and allowed us to begin to delimit the scope of the problem and initiate some management actions. And then Don Edwards National Wildlife Refuge was awarded an early detection rapid response grant from U.S. Fish and Wildlife for our 2023-2024 season, which was subsequently renewed for next year, 25, luckily. And this just really illustrates the importance of vigilance out in these marshes by knowledgeable biologists and stewards to protect marsh biodiversity, that we saw this threat and were able to respond to it. So here's the impetus for action why the South Bay Salt Ponds was able to get involved and help us with some interim funding, some pilot funding, was because they were planning to breach from mud slough into pond A19 to help with speed sediment accretion and marsh development at that restoration site. And there was quite a bit of this paspalum at mud slough, right where that green arrow is. And so it allowed them to shift a small amount of funding to get us started on figuring out what the level of the problem was and how we might tackle it. Paspalum vaginatum is a grass, but it is a turf grass. It is an escaped turf grass that forms these lawns, these dense lawns. Those mounds are very thick, some up to 2 feet or even more thick. Seems similar to Bermuda grass, but very salt tolerant. And you can see it on the lower left-hand corner, spreading in amongst bulrush and basically growing through its stolons, through its above ground runners into and through all that area. It does not grow down into the low marsh like Spartina so much, not out into the mud flats certainly, but it will colonize this middle marsh zone and dominate it completely. It was first reported by Reifner and Columbus in 2008, but they were referring to it as now widespread and highly invasive in estuarine wetlands in Southern California, and that it was likely unrecognized in the field for decades in Southern California. Quite a few sites in that article. Sean Allen confirmed that it was what we were already seeing in San Francisco estuary, that it's an early and effective colonizer of disturbed, bare, or ephemeral soil deposits, and once established, it can exclude indigenous species recruitment for many years. Interestingly, it rarely produces viable seed in significant quantities, so it must propagate vegetatively, so it's dispersed by fragments. Sold commercially in the turf industry in Southern California, and that's how it probably was introduced to the Bay Area. It probably washed into the flood control channels above Coyote Creek and got a start there. And was just, it looks enough like our native saltgrass that if people weren't really paying attention, they could assume that it was. And that's an area where we don't survey much for the Spartina because it's a more brackish area. So we're really not spending, the Spartina Project biologists are really not spending a lot of time that far up in the watershed, and that allowed it to get established with people not necessarily noticing it. So it's so salt tolerant, it can even be irrigated with salt water. So as you know, that's great as worldwide water shortages worsen, that there's a turf grass that can be irrigated with salt water that can be used in lawns and golf courses and other places. But unfortunately, it's not great for our tidal marsh and for the wildlife that they support. It's even been shown to survive pollution with crude oil. So, and an interesting thing that in these novel habitats, like say the South Bay salt ponds that are being restored, and they will eventually be tidal marshes, but they're sort of novel habitats in the beginning. USDA and RCS had stated about Paspalum that there was no environmental concerns. They just couldn't foresee this novel circumstance being invaded, and probably should have. We probably should have realized, but that's hindsight is 2020. So here you can see the invasive paspalum, or pava. I'll try to refer to it for the rest of the presentation. It expands by stolons, by these above ground runners, running into like our native gum plant there, our native grindelia, and our native bulrush. And basically you can see how it forms these large areas that exclude other vegetation. And grindelia is a plant that cycles. Gum plant is a plant that cycles, where it'll live for maybe, I don't know, 5 years or so, and it drops a lot of seed. And then once those mature plants died, it'll start to regenerate from seedlings and continue its, to own that space of its and provide that important vertical structure and habitat that it provides. Well, if you have a lawn of paspalum underneath that, it will not be able to do that, and will start to exclude and displace the grindelia completely and replace all that vertical structure that's really important for rails, harvest mice, and others. You can see it spreading into these areas, colonizing these benches along Coyote Creek. Just very good at is another, these noxious weeds are very good at capitalizing on disturbance, right? That's where they come in, that's where they make a presence, and then they start to spread within a site after that. Uh, to kind of touch quickly on the fact that we have basically two nodes. We have the infestation that's up in the Coyote Lagoon area and the flood control channels from which it maybe was first introduced. We have another node that's within A21 that to me appears like it was probably an independent introduction because the amount within A21 is not consistent with the amount that's in any of the surrounding areas, or there's not significant amounts between Coyote Lagoon and A21 that could have been like stepping stones that were really a lot of it was getting introduced back to A21. There's quite a bit there, so it seems to me that may have come in somehow by accident to A21 when it was early in its development of native plants, and it was able to spread quite rapidly throughout that area then. And then we have an upstream one in the map at the bottom of the page that's the upstream Alviso Slough area, where it may have been introduced in the boat launch at Alviso Slough because it's really just in that upstream segment and is absent from the bottom 2 miles of Alviso Slough as it goes out to meet Coyote Creek at the confluence there. And those points are not to scale, so it's very limited in some of those areas, and pretty extensive in others. So if you add up all the initial mapping events over the years as we delimited it and started to do some of our treatment because we couldn't address all of it at once with our limited funding, it probably was a baseline of about 10 acres by the time we started working on it. A lot of information in this slide, just kind of showing this, but showing the sites that we've identified, their treatment square meters, treatment cover square meters. Like Coyote Lagoon North there had already been treated in this 2023 mapping, so that's not its initial amounts, but you can see a lot of fairly small amounts scattered that we can still get on top of, get those down to very low levels, and then address these larger infestations and start to really reduce them from the meadows that they have become. I think I'm going to, in essence of time, just skip over the rest of that. We use Mazapir as we do in the Spartina project to treat them. A lot of airboat access for some of these remote areas in the bay. Some of them are extremely tricky, require very specific tide planning to be able to get the airboat into the areas as well safely, as well as have full exposure to be able to treat the plants, especially in the island ponds. This is an incredibly hard challenge to do. And what we found was that the Scarpus rainfall effect that I mentioned in my part of the Spartina project presentation really has affected this Paspalum too, which is not a huge surprise because you can imagine this is a more brackish plant species. It's doing great in the fully saline marshes of the San Francisco Bay, but it prefers to have more freshwater flushing. It's going to thrive more with some of the slightly more amenable conditions that rainfall will provide. And so what I found interesting was when I was out there this year, the top photo there is what I was seeing in 2023. And you can see a lot of open mud in that area. And this is an established infestation. It's not moving around quickly, right? Remember that the literature says the Paspalum doesn't produce a lot of viable seed. It spreads by a fragment and the fragment can root easily, but it doesn't produce a giant stand in 1 year, right? And then in the bottom slide, you can see some thick stands that I found this year when out treating in the same exact areas where nothing had been mapped or treated the year before. And it seems that effect of the rainfall, that there was already below ground biomass, root mass living in those areas, and that the rainfall really was able to stimulate that growth and allow it to show during those years. I don't have any other explanation for it, so it certainly is present at some significant levels. And this phenomena only happens really in those places where we had these large infestations. So that seems to be what the conclusion I'm drawing from it. So, but in A19, we found a lot more very small patches this year that don't seem to be spreading. Over the past few years, we haven't seen much spread, much new stuff popping up, and yet we did see quite a few of these smaller patches evident this year that weren't evident in the past. So, if we're lucky that we have the funding right now to get out and treat them and manage them, hopefully we can slow it down from really establishing in new areas and sending out propagules to new areas. And that's the adaptive management that we'll try to implement. So. And finally, Limonium. So Limonium is a sea lavender, and that the primary target Limonium that we have in San Francisco Bay is the Limonium ramosissimum, or Algerian sea lavender. First discovered, first recognized in about 2006, 2007 in Sanchez Marsh in San Mateo County, also in Marin. It tends to grow much more vigorously and produce the most seed in the high marsh and in what we call the estuarine terrestrial transition zone, or some people call the upland transition zone. So the vast majority is found above mean high water, although Lyra can thrive at the marshplain elevation and certainly along channel banks where it's more well drained. It will establish in intact marshes where there's a ton of seed being produced, and it can produce quite extensive stands along those channels and be a big problem. It can also grow as a dense substratum beneath the native marsh vegetation, a little bit like Paspalum. And then as it builds up critical mass, it will eventually exclude those native plants from there and displace them from their spot. Here's some beautiful photos from Ron Vanderhoff, one of the best weed photographers in California State, who lives down in Southern California, Orange County, I believe. He produces some beautiful photos. You see the pointed leaf tips of the lyra as opposed to the other invasive limonium, which is Limonium duriesculum, or European sea lavender, which has these more rounded tips, kind of looks more like a succulent plant. This is pretty limited in its distribution, mostly in Marin until we discovered, Calypsy's project discovered, an established infestation on Bear Island at Deepwater Slough, which was likely an independent introduction somehow because there was no other lidu anywhere else in the bay except for very, very limited amounts in Marin. So we're also starting to work on it there in Deepwater Slough the past 3 or 4 years. This is actually the more widespread limonium, invasive limonium in Southern California, where it's the bigger problem, and it is able to grow at a lower tidal elevation, from my understanding from my colleagues in Southern California, even into the Spartina zone. So, big concern. We want to not allow it to get out from its very limited distribution here in San Francisco Bay. Example of the more prevalent one, the one you're more likely to see around San Francisco estuary. On the left, Limonium ramosissimum, or Lyra, on the left, and then our native, some beefy, very healthy specimens of our native Limonium californicum on the right, that have bigger, thicker, wavier leaves, often have a lot of mildew on the leaves, and these plants are both boltings, which happens generally throughout all of May, and they start to flower into June and into July. So the project this is the project area this is a California Invasive Plant Council project. This was our 10th year for this project. We have a lot of partners around the Bay and a lot of people that are working on it independently, both volunteers as well as agencies and other groups. I'll mention that in a minute. But Calypsy itself received our first National Fish and Wildlife Foundation NIFWIF grant in 2015 for a pilot, and then we got several more grants over the years to help continue to fund this effort. It's a very shoestring budget, but so appreciated that they've continued to step up and help us, and hopefully they will again this year. Another grant application is in. We have also partnered with some of the county agricultural commissioners' offices to receive CDFA noxious weed funding from 3 to 5 counties, depending on the years. Where currently it's been 3 counties. Sites were mapped over time and added to the plate over time, and incorporated into regular treatment as the funding would allow. Peak infestation was probably about 25 net acres, which was quite a bit more treatment acres. Most of the top 10 largest sites have been reduced by 70 to 85% since 2016 to 2018. And this is a very, very data-driven prioritization of targets. There's a lot of invasive plants out there. We need to make our choices carefully as to what to invest in. San Francisco State Boyer Lab, their research was critical in our decision to take action on this specific plant. Its impacts to native marsh and its vigorous spread had already been well documented. U.S. Fish and Wildlife had also identified Lyra as one of the 3 highest concerned plant species in the South San Francisco Bay Weed Management Plan. The carpets and short rosettes of these 2 invasive limoniums cannot provide our native wildlife with that same vertical structure that I've been pointing out that is really important, that the natives provide, like Grindelia, like gum plant, that act both as refuge from predators, act as places where nesting substrate can be woven into nests for the Ridgway's rail, and for high tide refugia when these species are most vulnerable from avian predators, especially in the wintertime. manual removal can be very effective at this on Limonium, whereas the other two, Spartina and Paspalum, that's unfortunately not the case. With their rhizomatous plants, cover large areas, and manual removal is not a feasible option. It is for Limonium, and it's been widely used very effectively by local stewards. Can deal with nascent infestations, or they can local stewards can come in after an infusion of maybe several years of a higher level of effort with contractors getting reductions on some bigger infestations, getting them down to a manageable level where local stewards might be able to take up the mantle. One great example of that on a larger scale is the Hearns Head Park project in San Francisco, where the Port of San Francisco got a Restoration Authority grant. We're doing a much larger scale effort there, sea level rise adaptation and such. The Calypsy project had already been working for several years to reduce the limonium there, and then pretty much turned that project over to the Literacy for Environmental Justice folks that continue to implement a lot of manual removal and then outplant a lot of native plants to try to restore the habitat there. So that's a great model, and that model's there's several other examples of that model being implemented around the bay. And just want to shout out to the amazing work of volunteers around the bay on this plant, including at least one person who I believe is in attendance today, Martin Cooper, super volunteer from Save the Bay, Melissa Grush, who's also a super volunteer in my estimation who's protected the Hayward shoreline, and Roberts Landing from Limonium. Friends of Five Creeks, Marin Audubon with Judy Stalker as their lead, Golden Gate Bird Alliance, formerly Audubon, Greens at Work. These folks have pulled millions of plants. Martin and Melissa regularly report how many plants they're pulling. It gets into the tens of thousands quite quickly. These folks are extremely dedicated, and they count them. I don't know how they can count them while they do the work, but that's not within my skill set. But anyway, much thanks to them, because we don't have the resources to deal with all these things, and the work that the manual removal is just wonderful because it's very careful. You're catching every little plant. You're doing the least possible impact to the native plants surrounding, really allowing the system to thrive. So if we could do it all that way, it'd be great. It's just not feasible with the scale of the problem around the bay to do it all manually. But these folks are doing it, and they're doing yeoman's work for sure. We've had Civic Corps groups out there to remove the invasive limonium at the MLK shoreline in Oakland, and did some career trainings with them as well, as far as marsh ecology and the impacts of invasive plants, and some career pathway opening for them. And this is just a good example of how invasive limonium comes into a site. It colonizes these disturbance areas like these wave-swept shorelines. This is ideal marsh in the South Bay near the Dumbarton Bridge. Then it just dominates and basically excludes any native plant regeneration and owns that space. And it can also expand along these channels in years when the Grindelia cycles out. When the Grindelia blinks off, when it's not doing that, when it's would be regenerating from its seedlings, if the limonium is there, it'll just get a big boost, and then exclude the Grindelia in the future years. Like in this area of ideal marsh, where you can see the arrows, that's all invasive limonium along those channels where it's well drained. That's the initial infestation out there. So, and then just a couple more slides. The South Bay Salt Ponds Project has a lot of these levees around the bay. So it's no fault of theirs. This is what we're trying to do, is restore these habitats. But when you have this just above tidal elevation, which the limonium can thrive at, there's many miles that can harbor this invasive species that can act as a loci of dispersal to existing marshes, and can inhibit the future high marsh development as it finally happens at South Bay Salt Pond sites. And we want to have those native plants present at these sites, so that as the high marsh develops, they can come in and create that beautiful habitat, that vertical structure that we need to protect our native wildlife. And I refer to the lyra as tribbles from the old Star Trek episode, because it's like that. They're like these cute things, and then everyone's like, "Oh, what's the problem with the tribbles? There's no problem with the tribbles." But then there's so many tribbles that's Drew as Captain Kirk there, completely smothered with tribbles, or Martin Cooper is smothered with tribbles, and there's so many of them. And unfortunately, it produces so much seed, up to 130,000 seeds per square meter, according to Archibald and Boyer, at least in the upland transition zone. The seed bank tank life is quite long, probably 5 years. It has a virtually 12-month growing season. So, it can be an overwhelming thing. There's just so many of these tribbles, and we need to try to limit their spread, and then get ahead of them. You can see all the red seedlings there in that picture are just thousands more tribbles waiting to explode. And finally, the you can see on the left-hand side 2 generations of tribbles. You can see are the dead necromass from what we did treated the previous season. You can see the big plants, which are the plants that started to come up last year around July or August as we turned our attention to other things like Spartina. They grew throughout the year, and they produced these big plants. And then you can see the next cohort of tribbles coming up in the seedlings throughout that area. You can see we had very good success with our treatment, but the onslaught of the 12-month growing season and all those seeds is overwhelming. And it displaces the Grindelia, it displaces the Frankinia, and there's just a lot of these areas like this decommissioned road out at Knaptrack. It's just high enough for the Limonium to really thrive, and then it could spread to other marshes. And there are certainly other invasive plants out there that don't have programs working on them. And this is my one shout out to this plant, because it's quite a bad plant, and I don't think anyone has really been able to is able to do anything about it right now. Puccinellia marittima, or seaside alkali grass. It's a bunch grass. You can see in the photo on the left, it's smothering gum plant and dominating every single inch of both channel banks at a site at Deepwater Slough. And you can see there's acres of it there, and there's quite a few marshes where there's a lot of it. Ideal Marsh, Sanchez Marsh, Seal Slough, and others. So this gets back to the point that we really need to have ongoing stewardship throughout our marshes. We need to be out there seeing what new threats are coming in, deal with them early, prevent them to the extent possible, and with the goal of allowing healthy, resilient native marshes to mature and flourish. They'll have a lot more biotic resistance. They won't need the same level of maintenance and care throughout their entire existence, but to allow them to get started without these things stifling these bullies, these biological plant bullies stifling their growth, is really key. And with that, I will take questions.
Thanks so much, Drew. I like that term, biological bullies. That's great. But yeah, what a great presentation, really. And just big kudos to the Invasive Spartina team and the folks at Olufsen Environmental and all the people that are working on invasive species in the estuary. This is a lot of really hard work. I'll jump in. They only have one question so far, but I think people will have more questions once we get started. You alluded to drones, and a question from Marshall Dinowitz said, can the hybrid Spartina monitoring be done efficiently with drones?
Yeah. No, it can't be done efficiently with drones over a large scale, but it certainly can be incorporated. We're looking at incorporating it on some of the edges of the infestation, some of the outlier areas. We have very small, limited infestation in Sassoon Bay, and there's a lot of dense vegetation up there, including a lot of non-native vegetation up there, like non-native cattails and others, Lepidium. So using a drone to maybe pinpoint to fly over some of those inaccessible areas to find areas where we might then go in and monitor on the ground, then it could be very useful, and we're currently exploring that option right now. There's additional issues in just discerning between the hybrid and the native. So a lot, you saw that we do a lot of genetic sampling because some of them are so tricky. There are ones that are obviously quite obvious, but to really do a thorough job, it requires pretty much getting in on the ground, to actually get tactile with the plant, go through the morphological characteristics. Seeing it on eye level is pretty essential. So drones can be incorporated into a monitoring strategy, but they won't be able to do all of it. We also are looking at more remote sensing over the long term. So as the Spartina project, reaches its goals, we want to then have long-term monitoring, long-term stewardship as well. So something where we would be checking areas through remote sensing to pinpoint any suspicious amounts of Spartina growth, any new clones that could then be investigated and ground truthed on the ground, and at a much less cost than sending biologists out throughout the entire estuary.
So there's a question, I know this one can be really extensive, but maybe a brief answer on about a little bit about more about the herbicides you used and how effective they are.
So the herbicide used on Spartina and also the one that used on Paspalum is imazapyr. It's an acetylactate synthase inhibitor. It's considered it's in the category for EPA's categories of herbicides as practically non-toxic. So it's thought of in the conservation community as one of the safest options that can possibly be used because it's just very low toxicity, very low toxicity profile to even the foundational benthic invertebrates, mammals, human health and safety for the applicators as well. That is yeah, it's a very effective herbicide for monocots for grasses, and so that's why the literature suggested them as a possible option for Paspalum as well, and so that's what we had tried on Paspalum as well. The Limonium is a very difficult plant to kill. It's a broadleaf plant as opposed to the other two are grasses, so it's a dicot, not a monocot. And Vaslan is one of the best things to help us get a better efficacy on the Limonium, which is a triclopyr product, a low toxicity triclopyr product. Also another one that's kind of been formulated more recently. It's a more modern formulation of triclopyr that's a choline formulation that's it's a little bit yeah, a little bit better than the older formulations of triclopyr. And then we tend to mix that. We have to get two modes of action. We've often mixed that with the imazapyr as well. So.
So Sarah Pierce has a question. She works a lot in the marshes she's one of our cram specialists in the estuary. And she has a question, do short one-pager identification helpers for non-botanists exist for these species to help people be informed about how to identify them when they are out in the marsh?
It's a great question. We certainly could do that much more easily for Paspalum and Limonium. I've not particularly done that. There is the Calypsy website up in the upper right corner of this last slide where you can check out for some information on sea lavender and see a little bit more about that. It's pretty easy to distinguish, especially with a little bit of if you have the botany experience, it's pretty easy to distinguish from our native Limonium, and you're not likely to see the Lidu, so it's mostly the Lyra, the ramosissimum. So that would be a good one. The hybrid is difficult, so that we do a lot of because it's a hybrid swarm. It's a fertile hybrid that back crosses with itself. There are the obvious ones, but they're minimal over the course of the entire bay. There's a lot of these difficult to ID ones unless you have expertise, or even cryptic ones. So that would be a hard one to have a one-page fact sheet. We even do internal calibration trainings at least twice a season with our staff, that are all of our returning staff. And it is definitely not impossible to give some characteristics and some comparisons, but it's also somewhat regionally specific. We have Simon Gunner, the botanist I mentioned, that did our identification for Paspalum. Over the years, he set up these morphological ranges for which these plants grow in different areas of the bay. They're very different. So the inflorescence length has a standard range for foliosa of the ones we've sampled. Not exhaustively across all, but it has a morphological range. The different leaf characteristics, leaf width, leaf length, have these ranges, but they're very region specific across the bay. So we do have some of that data, but it's pretty deep, and it would be difficult but I think as the RMP blossoms that Don and I were talking about before the presentation, I think we can start to find a way to get some of incorporate that information into the little bit more skilled folks, the little bit more folks that have a little bit more botanical skills that are going out to do that work, get that information in their hands, and folks that are doing cram like you are, to have more eyes and ears out there, that at least something suspect could be reported, that then we could go follow up on.
Yeah, I was just going to follow up on that and say, I know in the past when I've been out in the marshes, I was able to take photos of things that if I was really concerned and send them to the Spartina Project. And I just wanted to see if that's something that people can still do if they see suspicious areas, I guess.
Yes, absolutely. And we Our website was ailing for a number of years, and we have a brand new, much nicer website with more of an interface to report things now. So that's right there in the center of the last slide there, www.spartina.org. So yeah, absolutely, and certainly just telling us where you're at, and coordinates are great, and then photos are great. Photos again, not to discourage, but photos can be tough. Even somebody who's seen thousands, tens of thousands of Spartinas, yeah, being there on the ground is it's really interesting how it's there's no the comparison. Like we go into a site, and a lot of invasive plant work is like this. What's different? What's odd? What stands out as odd? We go into a site and calibrate on what the native cordgrass looks like in that site. If it's if it's one where it's prevalent, the good mixed marshes of our bay where it's still prevalent, and then you're looking for what's different, right? And you have sometimes the morphological differences within the native cordgrass throughout the marsh. You kind of calibrate, this is this type, this is this type, this is this type. A handful hopefully is all you have. And then what is standing out that's not the tall one, the obvious one? Okay, that's a hybrid for sure, can see that from 100 feet away. But what are these subtle differences that we've documented in these marshes before, that are outside the morphological range, the normal morphological ranges, from our morphometrics, that kind of thing. So
Yeah, and sort of, I guess just following, it's important to kind of deputize the folks that are out working in the marshes to help these. This is such a great effort, and there's just a small number of people that are really making all this happen. And so anything that the greater public can do to help see, just mention when they see something suspicious even, will help with some of these early invasions in particular.
absolutely. And Limonium and stuff. And the volunteers are not, I was mentioning for Limonium, are not in any way, new to these things. They're super familiar with hundreds of thousands of plants. But I get reports from Martin Cooper and from Melissa Grush about areas, and we're able to follow up on them. So that's critical intel. In fact, Martin reported something he found at R4, the new Ravenswood Phase 2 part of the South Bay Salt Ponds project. Still limited infestation that we need to get on, that I was not aware of. So that is, Donna, you're absolutely right. That's we need to make the most of those things and find the best way to do that. Find the best way to have the information come in, that we can collate it and check it and make sure that it's useful for us. Yeah.
So we're almost at time. I think maybe one more question if anyone has a question. This has been a really great presentation with a lot of really dense, important information. So thank you, Drew, for like all the work that you've done to kind of in the past in the marshes, but also to help us understand what we're fighting against.
Oh
So
yeah, you're welcome. It is a lot of information, and I talk fast. I'm sorry, I
that's great.
apologize, but it is but I could give an hour presentation or an hour and a half on each one of these guys, so to fit them all in. But I want you to have a broad breadth of what's going on out there, and even see like the Puccinelli at the end out of that slide, even though it's even extra stuff. But we need to realize that we need to. It's just like in the old days, they said, the forests. Let's just set aside those forests, and they're going to be fine. And then we realized all the threats to forests and all the threats to forest fires and invasive plants and invasive insects. You can't just draw a political boundary around the marshes and have them actually be protected, or just breach them and have them develop. If they were pristine all around them, you could. But we're in an urbanized matrix with a lot of stressors, and we need to recognize that, and then and be vigilant, to protect the development of these marshes and protect their habitat for the long term.
Thanks again. And so thanks to everyone for attending. Just a teaser, next month we'll be hearing from Levi Lewis about who has synthesized some of the fish data in the South Bay, and he'll be giving a presentation on fish in the South Bay. So thank you for all for joining today. This recording of this meeting will go up on the website here probably in the next week or 10 days or so. So feel free to look for that. So yeah, thank you all.
Thanks everyone for attending. Appreciate it.